So the typical nerve cell has got a cell body, which is where you would find the DNA and all the machinery to read those instructions of the genes. And then what we saw in the animation at the beginning was this axon. So the axon is responsible for transmitting information to the next cell. And then these short structures here that tend to be short and thick and highly branched are the dendrites. And it's the dendrites that are receiving neighboring signals from nearby axons. And it's, a combination of 100 billion or so of these nerve cells put together in a very organized fashion that makes up your brain. So going back to that animation at the beginning, the action potential was that electrical gradient that moved down the axon and then terminated at the end of one synapse and made contact with the dendrite, and it's where that neurotransmitter was passed from the sending cell to the receiving cell.
Is it always a one-to-one relationship, or?
No, not at all, in fact. In the human cortex, you have about 10,000 synapses on one single cell. So there's a whole lot of information that is being pumped into any one neuron from multiple sources. And what the cell has to decide, ultimately, the output is binary. So you're either going to fire an action potential or not. There's no maybe a 20% action potential. It's yes or no. So from those roughly 10,000 inputs, that the dendrites are making a calculation based on the electrical potential of its membrane and saying, okay, I have enough excitability to create an action potential.
To the next value you want.
Yeah. And so to make it even more complicated is you have both excitatory and inhibitory neurons in your body. And typically you have a nice balance between those two. I mean, you could be philosophical and for a reaction is a reaction, but that's true. In fact, epilepsy is a classic example of the loss of inhibition. And the seizure is caused by, there's a temporary loss of the inhibitory neuron's action, and it causes a seizure. So not only do you have a multitude of inputs, some are saying yes, some are saying no, and the cell has to make a decision ultimately as to what it is. So just to give you some idea about how do researchers sort of understand some of these problems about nerve cells, and to add some life to that previous image. This is a time-lapse video of a rat brain cell developing in a culture dish. And so a friend of mine who studies this event, she spends her whole life on just this event. It's over in 18 hours. But there's a lifetime of questions to be spent on this point. But the idea is that nerve cells don't start out in this fantastic wired scenario. So as I showed you during development, you first have to lay out the basic architecture for the nerve cell. But then you have to have those axons extend and make the right connection with the right cell. I'll say a little bit more about that in the next slide. But this is a nice demonstration of how dynamic these cells are when they're developing, and the transition between a cell that's not so interesting before it has an axon to suddenly a cell that is going to go on to think great thoughts, presumably. So the point of that axon outgrowth and then looking for the right target is described in this little cartoon here, where, so if you think about neurulation, and after those cells have made the right migration to where in the body they need to be, now they need to start wiring together. And so the nerve cells undergo axonogenesis, And then that axon has to find the right target. So if we're going to wire our fingertips, those nerve cells have to grow out from the spinal cord and find the right finger and connect all that up with the brain, so I know that this is my finger. So there are a number of positive and negative signals. There are actually more negative signals that keep neurons sort of on the straight and narrow. But as these nerve cells migrate through the developing tissue, they are seeking positive signals that will allow them to wire one another so that they can ultimately make a person. So there are a couple of examples here of just how complex it becomes at the cellular level of a wiring diagram. So this would be an example of your olfactory neurons, or the neurons that are in your nose. And you have a set of olfactory neurons that are exposed in the environment, but then they have to make a whole bunch of connections to eventually make it into the olfactory region of your brain where you can process smells. This picture here is called a brainbow. It's using sort of tricks of molecular biology. You can make cells produce colors of all sorts. And so developmental biologists have used this mouse model to understand the wiring of the nervous system. So this is your hippocampus, or part of your hippocampus. And your hippocampus is a brain structure that's sort of right over your ear on both sides. And the hippocampus is important for sort of short-term memory. So memories come into the brain, they get processed in the hippocampus, and then they get distributed through the frontal cortex and through various other parts of the brain so that you can consolidate those memories if that's what you're indeed going to do. The hippocampus is also important for spatial learning. So, how is it that I don't get lost going to my office every day is because I have a spatial map, and I understand cues in my environment that tell me where they are. So a lot of those memories are encoded in the hippocampus. Important part of the brain. But the point is that it's got a very complex wiring system that needs to